Hello everyone and welcome to today's webinar, Tumor-on-chip 3D models for the analysis of programs, cell deaths, and tumor invasion. Thank you for taking time out and being here today. I greatly appreciate Dr. Samiei for accepting our invitation to present his experiences and achievements regarding tumor-on-chip. First of all, it is worth mentioning that this webinar is being held with a close collaboration between RASA and Omida Company. And now I would like to introduce today's presenters, Dr. Ehsan Samiei, with my pleasure.
Okay, can you see the screen?
Yes, of course you can see.
Okay, thank you very much. I would like to thank Rasa and Omida Fernand for taking this initiative and organizing this series of webinars, and I'm honored to be one of the speakers. So, as you may know, in drug discovery, 9 out of 10 drugs which successfully passed the preclinical studies, they fail in the clinical trials, and that causes the cost of drug development to increase significantly. The main reason behind this is the significant difference between the models that are used in the preclinical stages with the human physiology. So 2D culture plates and animal models are widely used for drug discovery. And so 2D models have limitations such as the lack of extracellular matrix, which causes the cell-cell communication and cell matrix interaction to be significantly different from human body. And also there is a lack of cell organization and tissue architecture. The animal models do not have such problems. On the other hand, there is huge interspecies differences between animal models and human physiology. So the differences in the tissue and the cell types causes different responses to the treatments and therapeutics. This demands more physiologically relevant in vitro tissue models to fill the gap between preclinical studies and the clinical trials. With that background, at the University of Victoria, we applied biomaterials and microfluidics for the development of 3D tissue models, specifically tumor-on-chip models. And today I'm going to present a few studies focusing on the development of the tumor model, their application for drug studies, including program cell death and also quantifying invasion. Then I'll show how such tumor-on-chip platforms could be great tools for the evaluation of other biomedical products, such as drug delivery carriers. And at the end, I'll present the further steps that we took to improve the quality of the tumor-on-chip models and increase their complexity. So through a collaborative research between University of Victoria and BC Cancer Foundation, we developed a tumor-on-chip model for mimicking glioblastoma, which is a grade 4 brain cancer. It is a very malignant cancer with a very low survival rate. Less than 5% of the patients survive more than a year. Using microfluidics and biomaterial, we mimic the microenvironment of the tumor. So as you can see in the schematic, the tumor-on-chip model involved a compartment where the tumor cells were encapsulated in biomaterial hydrogel in a 3D matrix. And then the adjacent compartment was cell-free biomaterial as representing the stromal region around the tumor. And the side channels, they were delivering the culture media representing the blood vessels. And in the first step, the model validation, a series of experiments for model validation were performed. As you may know, one of the main differences between 3D cell culture and the conventional monolayer cultures is the mechanism of molecular transport, where in 2D culture, the media is immediately available to the cells, but in 3D cell culture, the molecules have to diffuse through the network of the biomaterial and in the extracellular matrix of the cells. So to evaluate that behavior, we studied the diffusion of phytidextran through the tumor model, and we imaged it using fluorescence microscopy at different time points, and we observed a gradient-based pattern of diffusion of the molecule into the tumor compartment, which proved that the material transport is through the diffusion. Then the capability of the model in maintaining the cells viable and functional was tested, where U87 and U251, two main cell lines for glioblastoma, were cultured in the model for 72 hours. And as you can see in the graph and the image, more than 95% of the cells were viable after 3 days of culture. And the functionality was tested by staining the cells by GFAP, a biomarker specific to glial cells. And both of the cell types expressed positive GFAP after 72 hours of culture. One of the capabilities of our model was that quantifying invasion. And we were aiming to use this as additional parameter for this for studying the effect of drugs on the tumor cells. So in here in this image, we studied the number of invaded cells into the side compartment, the stromal compartment, and monitored it with 24-hour intervals, and we observed a reproducible pattern where the number of invaded cells increases over the period of culture. Then after validation of the model, we used it for drug testing. Temozolomide is an FDA-approved drug for treating glioblastoma. And simvastatin is a drug that is widely used for and is known for its cholesterol-lowering effect. And recently, it has been found to be effective when it's combined with other drugs for treating cancers. So in this study, we actually tested these two drugs separately. So the viability or cytotoxicity analysis showed a dose-dependent effect of the drugs on the cells. And as you can see, by increasing the drug concentration, the viability of the cells decreased. But the main observation in this test was that in comparison of the 2D culture and the 3D tumor-on-chip model showed that the model was more resistive to the drug. So the viability was significantly higher than the monolayer cell culture in both drugs. And that could be due to different reasons, such as the morphology of the cells or the molecular diffusion through the network, the biomaterial network. So morphology of the cells are shown here in the bright field, and also the immunofluorescence staining of F-actin filament of the cells. And you can see that the 2D culture, the cells are flat and attached to the surface, whereas in the 3D model, the cells have a tubular and elongated morphology. And the effect of drug on the cells is such a way that the cells lose their tubular morphology and become more round as the concentration of the drug increases. And that can be observed in the F-actin images as well. Then invasion of the cells in the side compartment was tested to see whether if it's a promising parameter to be used for analysis of the effect of drugs. So here, the series of images only shows the side compartment, only the cells that have invaded to the side compartment. And as you can see in the control group, there is a high number of the cells. And the side view image also shows that they are well distributed in the height of the model. and by increasing the concentration of the drug, the number of invaded cells decreases. And that has been quantified for both drugs, and a very reproducible and sensitive pattern has been achieved, which shows that this parameter can be used for the analysis of the drug efficacy. To understand whether this is only because of the cell death, or also it is because of the reduction in the invasiveness of the cell, we stain them, that the cells with vimentin, a biomarker which is one of the markers representing the mesenchymal behavior of the cells, and also is representative of the invasiveness of the cell. And as you can see, comparing the control with the treated groups shows that the treated groups show a lower level of expression of vimentin, and the quantification also represents that. And this shows that in addition to a reduction in cell viability, invasiveness of the cells also is affected by the chemotherapy treatment. The desirable cell death for chemotherapy agents is apoptosis, which doesn't cause inflammation. To evaluate whether the main mechanism of cell death is apoptosis, we stain the cells using Cleft-PARP, a marker representing apoptosis. And in the first step, a low concentration of the drugs was used, 100 micromolar TMZ, temozolomide, and 1 micromolar simvastatin. So both 2D and 3D models showed positive expression of Cleft-PARP, but the comparison showed a significant difference. So the monolayer showed a higher level of apoptosis than the 3D culture, which is in line with the viability results, and shows that the cells in the 3D model are more resistive to the drug, which is more physiologically relevant. And to ensure that apoptosis is the main mechanism of the cell death, we increased the concentration of the drug only for the 3D model, and we observed that the level of apoptosis was increased. Then to confirm that, we stained the cells with cleaved caspase-3 pathway that happens level prior to apoptosis. And similar observations were achieved. So for low concentration for 2D and higher concentration for 3D, and a similar level of expression of cleaved caspase-3 to Cleft-PARP was observed in this series, confirming that main mechanism of cell death is apoptosis. Glioblastoma patients and also glioblastoma is known to build resistance against treatment with temozolomide, and that's one of the main reasons of the low survival. One hypothesis is that autophagy is the reason behind that. Well, autophagy is a mechanism by which some organelles called autophagomes form inside the cell, and they act as a vacuum, so they collect damaged molecules and organelles from inside the cell, and then merge to lysosome to digest them and heal the cell. To study that, we stained the cells with LC3, which represent the autophagomes, and P62 is a cargo protein that is involved with the autophagy flux. So, and that represents where the level of autophagy gets to a level that autophagomes move and fuse into lysosomes. So, comparison of the treated samples with the control showed that both treated samples with TMZ and SIMVA showed a higher level of autophagy, more high number of LC3 puncta. But the main difference was that the colocalization with P62 was different. Temozolomide showed, as you can see here, the orange colored dots. The colocalization happened, which means that the autophagy flux was triggered, where the autophagomes fuse into lysosomes to digest the damaged organelles and molecules to heal the cell, which is hypothesized to be the reason behind resistance against temozolomide. Whereas the treatment with simvastatin, the co-localization was not observed, so the level was not significant when compared to the control group. And that, based on this, we concluded that autophagy might be the reason behind the resistance of glioblastoma against TMZ. In a separate study, we studied the combination therapy, simvastatin and temozolomide, with the goal of introducing simvastatin as a co-treatment therapeutic for temozolomide. And this study was also performed both in 2D and 3D. So the treatment were control individual drugs and the combination of them. And the morphology showed that the combination treatment, the morphology of the cells changed more significantly when compared to the individual drugs. So they are more round and they lost their tubular and elongated morphology. Then the viability was tested for both U87 and U251 cells, and similar pattern was observed. The number of dead cells for the combination therapy for both cases was observed when compared to TMZ or Simva alone. And the difference between the combination and individual drugs was significant. Simvastatin is known for its cholesterol lowering effect. And to study if the cell death, the increased cell death is because of that the cholesterol lowering effect, which is due to mevalonate changes in the mevalonate pathway or not, we tested the proteins that are involved in the mevalonate pathway. So we had similar treatments, individual drugs, the combination drugs, as well as new groups where the proteins involved in the mevalonate pathway into our treatments, and compare the results with and without those proteins. And so mevalonate and cholesterol are shown here, and two other proteins, FPP and GGPP, were also studied, which I have not shown here. And the difference, the comparison showed that the difference between the viability is not significant. And we concluded that mevalonate pathway is not involved in this extra cell death. Then apoptosis was analyzed. Here I've shown immunofluorescence imaging of Cliff Park, what we studied with flow cytometry and Western blotting as well. And in all three methods, we observed that the level of apoptosis was significantly higher in the combination therapy, which is a favorable result for battling glioblastoma. Then from the previous study, we hypothesized that autophagy might be the reason behind the resistance of glioblastoma against temozolomide. So here we studied the autophagy flux by staining the cells with L3, biomarker that stains the autophagosomes, and lysotracker, which directly stains the lysosomes. And so the results show that only the temozolomide treatment showed that the autophagosomes and lysosomes were co-localized, which means that they have autophagosomes merge into the lysosomes to digest the damaged molecules. And you can see the change in the color here. And that was not observed in the control group or the Simva alone or Simva and TMZ co-treatment. This was a good sign, and this showed that combination therapy, simvastatin and temozolomide, could be considered as an effective method for treating glioblastoma without building resistance against the chemotherapy. So as I mentioned in the beginning, the tumor-on-chip model was not only used for drug testing, but it could also be used for evaluation of other biomedical products, for example, drug delivery carriers. In a different study, we developed a shear thinning biomaterial for drug delivery where non-composite hydrogel was used, and doxorubicin as the drug, which is used for treating different cancers, including breast cancer, was loaded in the hydrogel to form a drug-loaded composite, and that could be used for intratumoral delivery or for embolization of the blood vessel feeding the tumor. So the biomaterial was tested. It showed great printability. The viscoelastic properties were tested, and it showed a great viscoelastic behavior. And also degradation and release profile of doxorubicin was studied, and the composition of the material was optimized to achieve a sustainable release of the drug over a long period of time, and the degradation of the material when it's injected inside the patient's body. Now, how the tumor-on-chip model was used to evaluate this drug. So to recapitulate what happens with the treatment with this shear-thinning biomaterial. So we injected the drug, so we made the tumor compartment in the middle of the device, and then injected the shear-thinning biomaterial in one of the side compartments. And that we hypothesized that makes a gradient of drug released into the tumor region. So first we studied the diffusion of the drug into the tumor-on-chip model. First, the free drugs dissolved in the culture media, and we observed that in less than 100 minutes, the concentration in the tumor compartment will be as high as the stores in the reservoir. So it reaches around 100% in less than 100 minutes. Whereas when we use the drug-loaded hydrogel in the side compartment, we observe a very sustained and slow release. And over 96 hours of release, we observed around less than 20% of the drug released into the compartment. And also it showed a gradient type of behavior, which is more relevant to what happens in human body. This was further shown by viability analysis, where the cells were stained with a live component of the live dead kit. And as you can see here in the two different time points, the viability shows that the site which is close to the drug-loaded hydrogel has fewer number of live cells, and in a gradient based, as it gets farther from the gel, the number of live cells increase. And the viability analysis also showed that the gel without the drug did not show significant cell death, which is great and shows that the nanocomposite is biocompatible, whereas the drug-loaded hydrogel at different concentrations showed significant increase in the cell death or in the reduction of the cell viability. Towards enhancing the tumor-on-chip and increasing its complexity, we considered developing tumor spheroids. As you may know, tumor spheroids are also considered as the 3D cell culture platforms where cells aggregate, and over a certain period of culture time, they start to secrete their own native ECM, which is more physiologically relevant, and therefore they are more biomimetic to human body when compared to the monolayer cell cultures. So we developed a microwell array, which was used for the formation of uniform-sized tumor spheroids. With a unique design and with an optimized design, the microwell was capable of distributing the seeded cells to the microwell in the perimeter of the array, and over 40 tumor spheroids could be formed with great circularity. The size and the circularity of the tumor spheroids was analyzed over different incubation times as well as seeding densities. So as the incubation time increases, the cells gain a better circularity because of the unique design of the microwell, and also their size increases. And then as we observed that seeding density could be used as a parameter for tuning the tumor spheroid size depending on the desirable size of the spheroid. Different cell numbers could be seeded, and as you can see here, the number could be, the spheroid size could be controlled by the seeding density. And also the circularity of the spheroids at different seeding densities was good, but improving with the higher number of seeded cells. Then we used these tumor spheroids and also the well itself, which is semi-transparent and can be used for real-time monitoring of the spheroids. We used it for drug testing, and we used doxorubicin again for the analysis of these tumor spheroids for drug testing. So here you can see the image of the spheroids on the platform itself, which shows a great potential for real-time monitoring of the spheroids. And this loss of integrity could be used also as a parameter for analyzing the effect of drug on tumor spheroids, which can be also observed in the confocal image when comparing untreated and the treated spheroids. Then the viability was studied and compared to the monolayer culture for both cell types, U87, which is brain cancer, and MCF7, which is a breast cancer cell line. And we observed that the viability in the tumor spheroids is significantly higher than the monolayer culture, which is more physiologically relevant. And this was observed for both cell types. Now, we have a more biomimetic tumor environment as tumor spheroids. And earlier I presented the Tumor-on-chip platform. Then we realized that combination of these two could improve the model and could form a more complex and more biomimetic tumor-on-chip model. So we encapsulated one tumor spheroid in the central compartment of the microfluidic model with biomaterial as the stromal region. And as you can see here, the tumor spheroid is in the tumor model. And other than cell viability, we observed that invasion of the cells from the tumor spheroid could be considered as a great parameter for studying the behavior of the tumor, and also use it for drug testing. The number of invaded cells increases significantly. Then as proof of concept, we use this the platform for drug testing using temozolomide and also doxorubicin, which is not shown here. And as you can see, the level of invasion reduces significantly. And the live dead analysis here shows that there is still live cells around the tumor spheroid in the 250 micromolar case. And that shows that the effect of drug is not only on the viability of the cells, but it also confirms that the effect of the drug is on the invasiveness of the cell as well. So it suppresses the cell invasion. Cleave PARP was again used to stain the cells to evaluate the apoptosis, and again confirming the results that I presented earlier. The low concentration did not show very significant level of apoptosis, whereas higher concentration, 250 micromolar, we observed a higher level of apoptosis, and this shows that this model is more biomimetic and more close to the human physiology. And another interesting feature of this platform was mimicking the diffusion-based drug transport inside the body. So here, instead of using free drug, we encapsulated doxorubicin in P9PAM microparticles and encapsulated those in a gel, which was delivered in the side compartment. So the drug-loaded P9PAM microparticle gradually and slowly released the drug, and we hypothesized that it forms a gradient of the drug concentration in the tumor compartment. And interestingly, when we observed the invasion of the cells, that pattern was clearly observed. So the lower side, which is the side that the drug has been delivered, there is no significant invasion, as you can see in both live dead and bright field images, and for both concentrations of the drugs. Whereas the opposite side, the level of invasion was significant. That proves the capability of the device for formation of such phenomena. That shows that even by increasing the complexity of the architecture, it would be possible to tune the delivery of nutritions or drugs to the tumor environment. And that showed also that invasion of the cells and metastasis could also be studied to analyze the invasiveness of the tumor cells. To summarize, because of the demand for more physiologically relevant in vitro models, we developed tumor-on-chip models, and biomaterials and microfluidics had very significant capabilities for developing such models. So biomaterial can be used for providing a 3D matrix for the cells, and that improves the cell-cell communication and cell matrix interaction. It can be used for formation of tissue and cell organization, and even by co-culturing and involving more, a higher number of the cells, more complex tissues can be formed. And also the mechanism of material transport is diffusion-based, which is more biomimetic. Microfluidic also was a great tool for recapitulating the structural organization of the tissues, and also by adding more complexity can be used for controlled delivery of nutritions, but not only nutritions and drugs, but also growth factors and cytokines, for example, for developing organoids and stem cell derived organoids. Microfluidic can be a great tool. And then the scalability of the system can be achieved using microfluidics for more systematic studies. And several steps can be taken as future directions, and which some of them have already been started, and several research groups, including our research group here at the University of Toronto, use different strategies to improve organ-on-chip models. So one way is to increase the to develop more sophisticated tissues, for example, by vascularization of the tissue, involvement of endothelial cells, and formation of the lumen, and also application of stem cell derived or patient derived cells to form organoids and develop more complex tissues. Such models can be used for personalized therapies instead of systematic treatment for all patients. Patient sample can be used in a short period of time and evaluate the response of the cells to drugs and their behavior, the invasion behavior. And then a combination therapy strategy could be developed for the specific patient with the goal of having better outcomes. Then scalability of the these tumor-on-chip models can be improved by using different methods. For example, more sophisticated bioprinting techniques, which more rapidly develop tissues in a shorter period of time, can be used for scaling up the production rate of the tumor-on-chip models, or in general, organ-on-chip models. Parallelization of the tissues could be a way, for example, such as 96 or 48 well plates, where multiple samples are in the same plasma and can be used for systematic study. several research groups are parallelizing such tumor-on-chip or organ-on-chip models, and they can be used as an array of several number of tissues. And other than that, the media delivery and therapeutic delivery, and in general, liquid handling can be automated, not only for delivery of the media and therapeutics, but also for taking the samples and for performing more dynamic readings for molecular analysis of the cells and the tumors. Here I finish my presentation, and I would like to thank my supervisor, Dr. Akbar at the University of Victoria, and I'll be happy to take any questions if you have.